Okay. A very good afternoon to all the viewers of this BAU Lecture Series 1. On behalf of Bihar Agricultural University, I welcome you all in the lecture topic that I'm going to deliver today. Before going to the lecture topic, I would like to introduce myself. I am Tirthartha Chattopadhyay. I have done my undergraduate and postgraduate studies from Bidhan Chandra Krishi Vishwavidyalaya in the Department of Genetics. And I have obtained my PhD degree from Indian Institute of Technology, Kharagpur. And I have joined this university, that is Bihar Agriculture University, in the year 2012. So I would like to thank our Vice Chancellor, Dr. R.K. Sohani, for creating this novel platform to share our research views, not only from the point of view of sharing our research thoughts or achievements, but also giving the scope to the students to learn from the research outputs that we obtain. So we have taken the topic of molecular breeding, which is really a very thrust area of research in case of crop improvement. So may I have the PowerPoint presentation, please? So during this presentation, I will be discussing about some of the molecular breeding activities that is being carried out by us at Bihar Agriculture University. I will request all the viewers to drop their questions or suggestions or any queries that they have in their mind in their chat boxes so that we can have those things printed during the presentation and I can get back to your answers. So, thank you. So this is the presentation, that is molecular breeding for tomato crop improvement. So in this lecture, the entire lecture is broken in 7 parts. The first one is a very brief introduction and novel idea about molecular breeding. The second one is a pictorial example of molecular breeding, how it actually works, which will be really useful for the students, I think, at undergraduate and postgraduate levels. Then about molecular breeding in tomato, which kind of issues we are dealing. Breeding objectives of our group that we have taken as the thrust area of research for location-specific cultivar development, as well as addressing some of the issues of national importance. Then multiple disease resistance breeding, which is feasible only through molecular breeding, as we are addressing through our program. Then breeding for improvement in fruit quality. And finally, I will give some of the suggestions regarding further readings in these aspects that will be basically helpful for the students associated with this program and who want to know about the molecular breeding in greater depth. So if we come to the first slide of the presentation, that is molecular breeding, we know that it's basically a conventional breeding. We follow the same strategies that we adopt for conventional breeding of a self-pollinated crop like tomato, but the selection procedure is assisted by some molecular markers. So why the selection is based on these molecular markers and what do you mean actually by the molecular markers? I will be clearing them. But to be very precise, molecular breeding is a conventional breeding method which is supported by molecular markers during the selection process, which makes the selection much more easier, labor cost effective, time effective, which can actually cut short the breeding cycle also. So before going to the molecular breeding, let's try to define the molecular markers. So the molecular markers are generally a stretch of DNA which is associated with a particular phenotype. That we know that if we go back to the Mendelian genetics, we know that each and every character is governed by some genes. Mendel proposed it to be a single gene. Later, we came to know that more than one genes can be involved in governing a particular character. So the molecular marker depicts us the variation in that very gene that is associated with a particular phenotype. For example, if I say that color development in flower, let's take, if we take a example that color development in flower, suppose it is a red color, then the red pigment is being synthesized by some biochemical pathways where a particular enzyme is playing a role, and that enzyme is basically a protein and being coded by a stretch of DNA, then if the change in the DNA sequence features can actually alter the protein, then the production of this red pigment is also going to be hampered. So we, in this molecular marker approach, we try to find out the type of variation which is existing between the red flower and the white flower bearing varieties, and we try to associate this variation at the DNA level with the phenotype. So that's why I have defined the molecular markers, that it's a generally it's a stretch of DNA that is associated with a particular phenotype that we call a trait. So why these molecular markers are really important? The first thing is, as we know, that any phenotype we see in the field or in any environment is highly influenced by the environmental factors, because the phenotype is the actual expression of the character, which depends upon the genotype, means the genetic architecture of the plant, and how these genes interact with each other and with the environment actually develops the phenotype. So whenever we try to find out a particular phenotype, then we are biased in such a way that the environment has played a certain role in that regard. Suppose if I take an example of plant height, it may be that during the preparation of the field, if my field is not properly prepared and in a particular patch the nutrition is much more available to those plants, then even with a different genetic architecture, a plant can get to some extent tall. So this kind of selections actually create problems to the breeders, because we cannot avoid the environmental effects. The second thing is, most importantly, the molecular markers, as they are DNA based, they are permanent in nature. So their expression pattern, it may vary still according to the environment, but the sequence feature that is associated with a molecular marker is going to be a permanent one. The third most important thing is, it can tell us about the zygosity, particularly when we use codominant markers. I have not taken any classification of the markers or dominant or codominant in my today's lecture, but to be very precise, a codominant marker can distinguish between a homozygous and a heterozygous individual. So we know that if we have selected a tall plant which is heterozygous in nature, then according to Mendelism, in the filial generation, this heterozygous tall plant is again going to segregate and going to give you 3:1 ratio of tall and dwarf plants. So this sometimes the breeders do not prefer. They want the fixed lines. They want to identify the lines which are homozygous at earlier generations, so that the desirable traits do not get the chance to segregate in the filial generations. Fourth one is, it allows simultaneous selection for multiple and difficult traits. This is very important. Suppose I want to select for a particular trait, which is a root trait, which is associated with a root, then the scoring of this trait is going to be really difficult, because to devise a non-destructive manner to score this trait is going to be really very difficult, because it will not allow me until and unless I uproot the plant and see the root architecture or some of the root traits. So when I uproot the plant is going to be destroyed. So if I can judge the root architecture from some of the DNA based features or sequence based features, then it's going to be a very useful tool for me. So the marker will actually allow us to see the unseen, even which is unseen from our naked eye. We can use it, we can use the molecular markers to predict, to identify that this particular plant or individual is going to bear this particular trait. And the fifth one that is very important is precise breeding. It can cut short the breeding cycle. So as I have already mentioned, that precise breeding means molecular markers actually influence the perfection of the selection of the breeders. All sometimes the students, they get confused that why we are focusing on molecular markers on some of the traits which are really very easily visible. still we are going for this marker test or something like that. So this actually give us the precision of selection. We are selecting not only on the basis of the phenotype now, but we are also giving equal importance, or you can say the more importance, towards the genotype. I can identify the homozygous lines which will be fixed in early generation, and I can select the filial generations for other traits, and I can simultaneously select for multiple traits also, that I have mentioned in point number 4, that multiple traits, this is particularly important, as I will be discussing later on, as these are particularly important for the traits which are self contradictory in nature. Suppose I want to develop a variety which is tolerant to drought as well as submergence, which is a very famous breeding objective in case of rice nowadays. So you can see that these two traits are self-contradictory in nature. So I cannot create the epiphytic conditions or the artificial conditions for the both traits in a same way, in a same pressure. So there can be chances of escapes. So in these conditions, the molecular markers are our tools, are our weapons that allow us to go for selection for multiple, difficult, and even self-contradictory traits. So let me introduce the molecular breeding. This is particularly useful for my students, undergraduate and postgraduate level, that how the molecular breeding actually works. So I've taken an artificial and arbitrary example, that suppose I give that A gene is giving red colored tomatoes, and it is a dominant gene, where the loss of function small recessive allele gives you yellow tomatoes. Now suppose that you have a particular breeding program where your objective is to breed for yellow tomatoes. Now the line that you are having in yellow tomato is not a good yielder. So what we do actually, we cross this line with a good yielder, and we want to take the yellowness from our variety, and we want to combine both the good traits. Suppose if yellowness is my desirable trait, then I want to have yellow tomatoes with good yield. So if I am crossing a plant parent 1 which is capital A bearing red tomatoes, and I have crossed with my parent 2 that is small a which is yellow tomato bearing line, then the F1 plant that is having a heterozygosity capital A small a is going to be red fruit bearing, because I have mentioned that A is a dominant allele. Capital A is the dominant allele of this gene. So in the next filial generation that I will get after selfing of this F1, there it will follow the simple Mendelian inheritance, that is capital A, capital A small a, and small a. These three genotypes will be produced in 1:2:1 ratio, and the phenotypic ratio is going to be 3:1, 3 red lines with 1 yellow line. So now remember, I have taken the trait which is expressed only in the fruits. It means that if I have 1000 F2 plants, then until and unless these 1000 plants give their fruits, I cannot select which plant is going to give me actually yellow colored fruits. But if I can have some molecular markers for the A gene, then the picture is something different. Let's look at, suppose I have developed a molecular marker where the capital A gene gives me a band of suppose 300 base pair after doing the polymerase chain reaction, that is PCR, and small a allele gives me the 200 base pair band. So these two bands are now allele specific bands. For the capital A allele I get a 300 base pair band, and for small a allele I get a 200 base pair band. So if I do this genotyping through the molecular marker based assay, then this picture tells me that parent 1 is producing 300 base pair band, parent 2 is producing 200 base pair band. Now this is an example of a codominant marker, means in the F1 both the bands are going to be present. As I know that the F1 contains the characters from both the parents, so if this character is this marker, if I consider as a character, then this character will also be present in heterozygous condition, both the bands will be present. Now in the F2, if I take the leaf sample from each and every seedling, isolate the DNA, and do the same marker analysis with the genotyping, which is very easy to do, then in the laboratory within 3 hours, I can come to a picture like this, where I know that plant number 1 is producing a band of 300 base pair just like parent 1, like 3 is producing both the bands like F1, 5 like F1, 6 is like parent 1, 9 is like parent 1, 10 is like parent 1, 7 and 8 are like F1. So I know that all of these plants are going to bear red color fruits, as I have mentioned in this inheritance pattern of this gene. But I have identified two plants, plant number 2 and plant number 4, which are producing the band size just like the parent 2, which is bearing the yellow fruit. So out of these 10 plants, if my breeding objective is focused on development of yellow colored tomato lines, so I can discard all these 1, 3, 5, 6, 7, 8, 9 and 10 number plants, and I can select plant number 2 and plant number 4. Now, the added advantage is you see that plant number 2 and plant number 4 are homozygous. They are not having any gene like the parent 1. This band is absent. So in the filial generations, in F3 or F4 or further advanced generation, they are all going to be true breeding lines. Always I will be getting only yellow fruited lines. So if I have selected some of the heterozygous plants, then the story will be just like the same. In the F3 generation, I can again go for a selection for those plants which will bear the yellow fruited tomatoes on the basis of genotyping. So this is a very simplified explanation of the molecular breeding program, as we see that why this system is important and how it can really cut short our labor. So you can easily understand now, out of these 1000 plants that I have mentioned, theoretically the probability is 1 by 4. So there are only 250 plants which are going to bear theoretically yellow colored fruits. So if at the seedling stage I can identify that these are the 250 plants which are going to give me yellow colored fruits, which is my breeding objective, then I need not to take the burden of the rest 750 plants. I can cut short them from my breeding program. So naturally the land required to grow these plants will be very less. Previously, I was transplanting 1000 plants, then I was selecting for some of the yellow fruited lines. But here, on the basis of marker based genotyping, I know that these 250 plants are going to be my targets. Now, the question comes that how perfect the genotyping is going to be. So to this example, what are the assumptions? The assumption is that A is the only gene that is involved in this color development, and the marker that you have used is a perfect marker. Means all the plants which are carrying this 200 base pair small band are going to give you yellow fruited lines. So often the breeders, when they develop the markers, they test this kind of things, that whether the trait and the marker is following 100% correlation or not. If you are using a gene based marker which is based on the gene itself, then it is obvious that it is going to give you a complete trait marker association if it is a trait that is governed by that very single gene. But if multiple genes are involved, then the story will definitely become more complex. Not only that, there are certain markers which are not gene based markers, but they are associated markers. Their position is a little bit distant from the gene of interest. So in these kind of markers, we call them the linked markers. There is a chance of recombination between the marker and the trait gene. Means, as simple as if I take the example of A and B gene, there is a chance of recombination between A and B. So if the distance of the marker from the gene is more, then the chance of recombination is also more. And if the distance between the gene and the marker is less, then we call it a tightly linked marker. Then the chances of recombination will be less. And recombination means I will get some of the plants where a marker band is present but the trait is absent, and I will also get some of the plants where the marker band is absent, but still my trait of interest is present. So this happens with linked markers, where recombination takes place between the marker and the my gene of interest. But obviously the breeders, they look for perfect markers, which are gene based markers. Often we call them the functional markers. So in our group also, we have the objective to develop some functional markers that can really predict the phenotype in 100% correlated manner. So with this basic example on molecular breeding, I will go forward to give you a brief introduction about this crop, that is tomato, on which I am working on. So the scientific name is Solanum lycopersicum. It is a diploid species having 24 chromosome number, having a big genome size of 950 MB with approximately 35,000 genes, and it is a model dicotyledonous plant, basically for studying the fruit ripening related traits. It's a self-pollinated crop. So all the breeding methodologies that are adopted for self-pollinated crops is applicable for this crop also. And India is a good producer of tomato next to China. And within India, Andhra Pradesh is the leading producer, and Bihar ranks fourth. Now, the problem associated with the crop is two types. Means the first problem is this crop is such a crop which is sold from 0.5 rupees a kg to 80 rupees a kg. Means it has a huge market price fluctuation, which is indicative of its particular season of production. If you go to the market nowadays, or at least 1 month back, tomatoes were sold at suppose 1 rupees a kg or 2 rupees a kg at maximum. So the farmers don't get the price out of that. But just after a few months, in the month of July, when you will go to the market, you will see that tomatoes have become much more precious. Now the consumers want to purchase tomato even at 80 rupees a kg. And even the price rises, particularly in the month of November. Again, it reaches up to 50 or 60 rupees a kg, as I was seeing the data from different ICR sources. So this kind of a price fluctuation is very much associated with tomato, and the consumers, they prefer tomatoes for different purpose. Means if I want some culinary tomatoes, I will need juicy tomatoes. So if I need some processing quality tomatoes from which I can prepare the ketchup, or I can use it as a salad, then I need fleshy tomatoes, the tomatoes which are containing less juice and more flesh. Because the processing industry, during preparation of the ketchup, they boil it. So if the tomato contains more amount of juice or water, then they will require more energy to evaporate that to prepare the puree. So they look for fleshy tomatoes. So what I want to actually mention, that this is the crop where the size, color, weight, storability, flavor, taste, firmness, everything matters. So the first bottleneck was that the proper climate you require. Second is, being a solanaceous crop, it is attacked by a huge number of insect pests and diseases. And the third is, you have to retain or you have to maintain a particular quality standard if your tomato, if you want to fetch good price for your crop for the farmers. So let's look at the major diseases that occur in tomato. The first one is a late blight caused by Phytophthora. Second one is leaf curl, the most devastating disease of tomato, caused by a vegomovirus, which is transmitted by whitefly, and it can cause up to 100% of yield loss in course of the crop. And uprooting and burning is the only measure when it has occurred in your field. You can check the transmission by whitefly by application of some of the insecticides. But if the transmission is already there and the crop is infected, then uprooting and burning is the only possible measure that you can take. And the third one is an uh, invisible disease that we often don't see, that is root knot nematode. If you go to the field in the first half or in just after the morning hours, if you find some of the plants which are droopy in nature, which are showing stunted growth, you can find that there are some root knot nematodes. So you look the diversity of the diseases. The first one is caused by a fungus, second one is a viral disease, and third one is a nematode. So naturally, if you want to breed for this type of diverse diseases, there is no single epiphytotic condition that you can create. So until and unless you have very good molecular markers that can actually predict that, okay, this line is going to give you resistance and this line is going to give you susceptibility, then breeding for multiple diseases is really very, very much challenging in case of a crop like tomato, which is affected by diverse type of pathogens from fungal, viral, nematodes, etc. So I will explain the fruit color variation also, which is a very determining factor for tomato production. You know that fruit color is actually the effect, additive effect, you can say, between flesh color and skin color. So the skin is the tomato peel that we get, and the skin generally contain a flavonoid named as chalconaringenin or naringenin chalcone. This is a yellow colored compound, and this yellow colored compound gives the yellow skin to tomatoes. So when the normal red flesh of tomato is behind this yellow skin, we get sharp red colors, bright tomatoes. But when the same red flesh color is under the colorless skin, where there is no naringenin chalcone, we get pink tomatoes. Nowadays, in certain markets, the availability of pink tomatoes are there. We also have some of the lines with pink colored fruits, and they are really attractive for the consumers also. So what I want to mean actually is that the skin color is also a very pivotal, plays a very pivotal role in ultimately determining the fruit color. And this is from one of our publications that we fetched in Plant Cell Report in the year 2021, where we identified the different mutant alleles or the point mutations that actually cause variation in tomato color. You can see that IDI is a gene which, where a mutation, if a mutation takes, then the tomato is basically devoid of any pigments. There may be mutation in the PSY1 gene, phytoin 1 gene, which will cause abolishment of major pigments from the tomato fruit and cause a yellow flesh. You can see that there can be a loss of function mutation in carotene isomerase gene, which can lead to production of tangerine colored tomatoes. And you can find that some of the gain of the function mutants can actually increase the delta carotene content in tomato. This is known as the delta mutant. And in the and mutation in the CYCB gene, they can give again color variants where the old gold and old gold crimson, these two mutants, they contain a huge amount of lycopene, whereas the gain of function mutation of CYCB gives a tomato with beta carotene content, high beta carotene content, and hence this mutation is termed as the beta mutant. So this is the only mutations I have mentioned in the carotenoid biosynthetic pathway, but there are several other regulators of this carotenoid biosynthetic pathway which actually add up to different color variation in tomato. Now, why we are actually interested in color variations in tomato? For one example, let's take the example of Critso, which converts the prolycopene to lycopene. Now, what recent researchers have claimed that lycopene is very beneficial to our health, but lycopene is very less absorbed by the human body. On the other hand, its precursor, that is prolycopene, has all the health benefits which are associated with lycopene itself, but the absorption of prolycopene is much more. So you look, this Critso mutant is actually a beneficial mutant for us, because it will stop the lycopene formation. It will give tomato fruits which are enriched with prolycopene. So actually this prolycopene's absorbance will be higher in human bodies. So with the Critso mutation, we can actually improve the nutritional quality of tomato. So here are certain more variations in tomato color. One of the variation that has been breeded by actually Dr. Hajra and his group at BCKV, where the AFT and DG, these two mutant alleles have been stacked to develop the purple tomato, which the institute has claimed to have a very pivotal role in fighting against cancer, because anthocyanin is an anti-cancerous compound, as we know. So this is another indigo rose colored anthocyanin rich tomato where AFT and ATV, these two mutant alleles, have been stacked. And there are more color variants nowadays being added in tomato, where orange brown colored tomato rich in phytonutrients and other health promoting nutritive compounds has been developed. They are not developed through any transgenic approach. They are non-GMO product. Only through molecular breeding and mutation breeding this kind of color variants have been added. So this is a beautiful work that has been recently published, that San Margiano is a very famous tomato variety for processing type. You can see wild type. And these rest HP1, HP2 and all other, they are isogenic lines of this San Margiano. And they have been developed through molecular breeding by incorporation of particular mutant alleles in this background of San Margiano. So you can find out a beautiful panoply of colors where all the mutant alleles have been brought in a single genetic background to have the color variants in isogenic form. So the shape of the tomato is again going to be a very important factor for its selection. As I have already mentioned, that juicy tomatoes are generally preferred for culinary purposes, whereas the processing quality tomatoes that you need, we generally prefer them to be a thick fleshed. They should contain less amount of juice, like that. So several genes, they control the fruit shape in tomato. One of them is ovate, which is a negative regulator of fruit elongation, because in general the wild type tomatoes are round in shape. So you can see that when the domestication process of tomato actually started, then it added up to different fruit shape variation in tomato. And nowadays we have a lot of cultivated tomatoes that vary in their shape. We have pear shaped tomatoes, we have ox heart shaped tomatoes, we have blocky tomatoes, we have flat, round and flatty shaped tomatoes. So there are four genes basically that play a pivotal role in regulating the tomato fruit shape. They are FAS, LC, locule number controlling gene, ovate, and SUN. So ovate is one of the very important regulator of fruit elongation in tomato. When ovate gene is functional, the tomato fruit is generally flat in shape. And flat shaped fruits, they generally contain more amount of juices. So they're preferred for culinary purposes. On the other hand, the elongated fruits are really good for processing, because recent research works have found that the mutant allele of ovate that gives you elongated fruit is also associated with fruit firmness, which is a trait that is required for long distance transport of tomatoes. If we think about the processing of tomatoes and we want to develop processed products of tomato, they need to be carried from one place to the other place, and they must support this transportation, long distance transportation. I can mention about the flavor shaver, the genetic engineering tomato that was introduced previously, where the fruit firmness was mainly addressed through genetic engineering related technologies. But through integration of the ovate mutant allele, we can have this fruit firmness in our cultivars in non-GMO approach through through simply molecular breeding. So this is the fruit shape variations that are associated with ovate mutant. You can see that they are basically elongated type fruits. Hence, the selection of ovate allelic status is very much important in tomato breeding programs, because as I have mentioned that this crop faces a price fluctuation. So naturally we have to breed for some of the varieties which are really very good for processing. So this ovate allele is going to be a very good allele that we can actually introgress in our lines to have farm fruits that can have good processing attributes. So on the basis of this information, our group at BAU, we are working on some of the objectives. The first one is stacking of different disease resistance allele in tomato genotypes, and we focus mainly on three resistant alleles. First one is TY3 that gives resistance against tomato leaf curl disease, MI1.2, the gene which gives resistance against root knot nematode caused by Mil.2 incognita, and the pH3 allele, which is a horizontal disease resistance allele, giving much more resistance against different strains of Phytophthora infestans causing late blight in tomato. We are also working with some other disease resistance allele, but our major focus is on these three genes. The second one is identification of fruit color mutants at molecular level and development of DNA markers. Because here the DNA markers are going to play a very important role, because you know that if you find a yellow tomato, it's very difficult to identify whether this is a PSY1 gene mutant or this is a mutant with high beta carotene content. So we need to develop some of the identification technologies through which we can say that which causal mutation is actually responsible for fruit color variation in our lines. The third one is identification of fruit shape mutants and development of DNA markers. This is particularly we are focusing for developing farm fruited lines with good processing attributes. Fourth one is molecular breeding for fruit quality traits using marker assisted selection, and ultimately combining the fruit quality with disease resistance alleles to get superior tomato genotypes. So here is one of our works that where one of my present MSc student is working on. So uh, we first tried to pyramid the two genes, that Ty3 and Ph3. For that, we first screen for the presence of the Ty3 resistance allele through gene-based marker, that is Ty3 SCAR1. This is a representation of a few lines, a few tomato genotypes, where the Ty3 resistant allele, which is of 519 base pair, is identified, and rest of the lines are carrying the susceptibility allele. So one of the donors for this Ty3 is Vir-TOLC32, and you can see that CLNB, which has been identified as a good material for heat tolerance in tomato, it lacks the Ty3 resistance allele. In a similar manner, a mutation in the late blight resistance gene PH3 makes it non-functional. So the functional gene, we track it by the marker PH3 scar that has been developed by different workers, and it identifies this deletion, whether this deletion is present in the gene or not. If the deletion is not present, then it is a resistance allele, it is a functional allele. But if the 22 base pair deletion is present, then obviously this is going to be a non-functional allele. So this is the allelic status of our different genotypes. You can see that in CL and B, it has produced the 150 base pair susceptibility allele, whereas the TOLCV32 genotype has produced the resistant allele, that is 176 base pair. So in a nutshell, CLNB, which is a good material for heat tolerance, is lacking both the Ty3 and Ph3 alleles. On the other hand, the Vir-TOLCV32 genotype, which is used as a donor for leaf curl resistance in tomato, carries both the Ty3 and the Ph3 resistance alleles. So what we did, we made crosses between CLNB and Vir-TOLCV32. We identified the F1s through heterozygosity testing, and we used 296 F2 plants for genotyping. So this genotyping was first done for the Ty3 resistant allele. So you can see here that the lines which are having the lower band, they are carrying the susceptibility allele. The lines which are carrying both the bands, they are heterozygous in nature, and the lines which are carrying only the upper band is the resistant allele containing lines in homozygous condition. So we identified the lines and marked them by the green lettering. You can find out here that we identified in total 49 plants out of the 296 plants which carried the Ty3 resistance allele in homozygous condition. So this allele is now fixed in these 49 lines. In their filial generation, all of them, all the progenies are going to have Ty3 in homozygous condition. So this gene is fixed. So next, we screened only these 49 plants which have contained the Ty3 resistant allele in homozygous condition for their allelic status at pH3. So here you can see the green color shows you the presence of pH3 allele in homozygous condition. So out of these 49 lines, we have identified 11 plants which are having pH3 resistance gene also in homozygous condition. So now, in a nutshell, if we see about the pictures, this is the cultivar CLNB lacking the Ty3 resistant allele as seen by the P625 marker. This is the Vir12CV32 line producing the resistant allele. You see that in the F2 generation, all the 11 plants that you have identified, they are homozygous for the resistant alleles. So they are fixed. So we have been able for early generation fixation of this allele. So we also checked this 11 plants with gene based markers. We got almost identical result except one plant, that is 43, which has shown heterozygosity on the basis of the gene based marker, which says that P625, as have been depicted in some literatures also, that this is a linked marker and there is a chance of recombination between the marker and the gene, which can cause this kind of anomaly. But for rest of the 10 plants, all are positive for the Ty3 resistance gene when tested with the linked as well as the gene based marker. And this is the picture of the Ph3 gene identification in resistant 11 lines. So in a nutshell, what we get from this breeding program is we carried out genotyping of 296 plants in F2 generation. We identified 49 plants which are homozygous for the Ty3 locus. At the same time, out of these 49, we got 11 plants which are homozygous for both the Ty3 as well as the Ph3 locus. So now, for the students particularly, I will try to explain that look at the impact of marker assisted breeding, that out of these 296 plants, but we have selected only 11 plants, and truly out of these 11 plants we have selected again only the best 5 plants on the basis of their morphology. So you look what amount of precision the molecular breeding can actually give you. Out of these almost 300 plants, you have now selected only 5 plants in the F2 generation, and you know which plants you have selected, and you know that these plants are not going to segregate for the Ty3 and Ph3 resistance alleles. These two resistance alleles are now fixed. All you want to do in the next generation is selection on the basis of good morphological traits. And you know all these plants are going to have the resistant alleles for the Ty3 as well as the Ph3 gene. So we tried another approach where not two but three genes we tried to stack through molecular breeding. You know, as the number of the genes, target genes to be stacked increases, the workload actually increases, and you need to screen more and more genotypes. So we first developed a breeding scheme using 4 parents which are having these 3 disease resistance allele, that is Ty3, Mi1.2 and Ph3. Definitely there was no single parent which contained all the 3 genes, but in some of the parents we got Ty3, in the other they contained Mi1.2, in other one it contained Ph3. So we went for 4 parental cross. We crossed A into B and we crossed C into D, and then the 2 F1s were again crossed. So in the double cross F1 population, we screened around 200 plants. And look, out of these 200 plants, we obtained a single plant where the Ty3 loci is fixed, but Mi and Ph3 loci is in heterozygous state. So we selected this single plant only because we know that in the next generation, when recombination will take place, we will get some plants which will be homozygous for the Mi as well as the Ph loci, and the theoretical probability is 1 by 16. So we took the seeds of this plant and we screened around 120 progenies of this single plant, and we identified 8 plants. So look again, what I want to mention for my students, that out of these 200 we selected one. Out of this one we again raised 120, and from this 120 we selected only 8. So the precision of our selection is actually increasing, and these 8 plants, all these 8 plants are homozygous for all the 3 loci. This is one of the work that is being carried out by one of our PhD student. So these plants seed we are now harvesting, they will be advanced in the next generation. So you can see these are the 11 plants that we have developed from these 4 parents: IHR 2614, Kashi Chayan, CLNB, and Vir12 You look for the Ty3, one is lacking, having. For the second gene based marker also, lacking Ty3, having Ty3, lacking Ty3, having Ty3. For Mi 1.2, only one genotype IH at 2614 is having, rest of the three is lacking. For the pH3, IH at 2614 and CLNB is lacking, whereas Kashi Chayan and VR2LCV32 is having the resistant allele. But our 8 plants, they are containing all the 3 desirable alleles in homozygous condition. So merely selecting 200 and 120, almost 320 plants, we can achieve the lines which are having 3 resistance genes stacked together, and they are going to serve as our pre-breeding lines, which are very much important, that when we try to transfer all these genes in a particular cultivar, then these 8 plants can be used as pre-breeding lines that can donate all the 3 resistant alleles at a single crossing event. So breeding for improving lycopene content, as I have already mentioned, that the fruit color is and nutrition content is a very important aspect in tomato breeding. So here one gene, that is SL date 1, deethylated 1, a single point mutation in this gene actually causes a huge increase in tomato lycopene content, and the mutant is known as HP2, high pigment 2 DG, dark green. The fruits, the immature fruits, are dark green in color. They mature. When they mature, the red color, the lycopene content is very high. So the problem associated with the identification of this gene at DNA level is a little bit difficult, because as I mentioned, like previous markers, it is not so easy, because here a single A residue is changed to T residue. It's a single nucleotide polymorphism, SNP. And you know that to identify the SNPs, you require specialized equipments, and you require some high cost fluorescence based chemicals to detect this single nucleotide polymorphism. Because if you do a PCR in both the wild type and mutant, they will give the same size of band, because the band length is not changing, rather the sequence at a single position is changing. So there is a procedure known as tetraprimer amplification refractory mutation system, through which PCR based primers can be designed to even identify the particular point mutation. So we developed some T-arm strategy, and through this strategy we don't use 2 primers, rather we use 4 primers in a optimized ratio, so that we can identify that which one is a wild type and which one is a mutant type, which where these two genotypes at this gene are differing for a single nucleotide only. You can see A gives you the wild type and T gives you the mutant, and look at the impact of the single nucleotide change in tomato fruit. So this is our study, and we have been fortunate to publish it in recently in 3 Biotech in the month of March by one of my MSc students, that is Arnab. So this work has been published, and you can see that these markers that we employ, they can identify the wild type by the presence of 2 bands, a band of around 195 159 base pair, and a common band that is of around 297 base pair. So this 297 base pair band is present in all, whether it is a wild type or it's a mutant. Now all the wild types will give you a 159 base pair band, but if it is a mutant where A is replaced by T, then the band size will change to 195 base pair. So naturally this is a codominant marker. We identified our mutant line that is containing this dark green mutant, and we also tested our F1 plants. You look, this is the wild type having the 297 and 159 base pair band. This is the mutant having the 297 and 195 base pair band. And in all the F1 plants, you can see all the 3 alleles, just like the codominant marker, where the alleles are, where the heterozygosity is revealed by the presence of both female and male specific bands. So we carried out our F2 genotyping also, and in F2 genotyping we have already identified 11 plants which are homozygous for this dark green mutant allele. So we expect to get good amount of lycopene content in these 11 lines. These are now in field condition. So the strategy that we have taken is the point mutation in the det1 gene. We developed perfect DNA based marker. We validated the marker. We adopted marker assisted selection in segregating population for the identification of better segregants. So continuing to this fruit quality improvement, as I have mentioned, the shape of tomato is also a very important feature regarding its quality. So another gene known as SL-ovate, this is also a negative regulator of growth. So this ovate gene, whenever, as I have mentioned, whenever it is mutated, it gives you an elongated fruit type, which is very important for processing, because ovate mutation is not only giving you a elongated fruit, but also giving you the fruit with high fruit firmness, which is a desirable character for processing industries. Here also the story is almost same. Only a single nucleotide change from G to T causes this mutation. So a single nucleotide changes and a flat tomato is converted to an elongated one on the basis of the ovate gene. Other genes also play a role, but ovate is also having a very pivotal role in determining the tomato fruit shape. So we, in a similar strategy, using just as I have explained for the previous one, we developed four primers that can actually identify the single G2T mutation. And here is our result. You can see in all the wild types and mutants, you get a 246 base pair common band. In case of the wild type alleles, you get a 161 base pair band. Whether Wherever there is a mutation present, you get a 139 base pair band. So these two are our mutant lines, as I have mentioned. One of them is IHR 2614, and the second one is Punjab Chuhara, a very famous variety that is taken for processing quality tomatoes. And we used these markers in our F2 population of around 200 plants. We screened more than 350 plants, and we identified the plants which are having elongated fruit shape, and also identified the plants which are not having the elongated fruit shape, means they are flatty type. So you can see that in all the elongated fruits, you find the banding pattern to be as similar as the mutant line that was used as a female parent. So from the DNA profile of the seedling, you can predict that, okay, this line is going to bear an elongated fruit type. So if you are focusing on developing processing quality tomatoes, you can genotype your segregating materials. You can find out the mutant allele specific banding pattern, and you can promote only those plants in the field condition that will reduce your labor cost, your time and resources, and will give you a precision in selection. On the other hand, in case of non-elongated types, they were basically like the wild type. In one plant only we found the heterozygosity, which is quite natural as this mutation is a recessive mutation. The heterozygous individual is also going to bear the flat type of fruits. So this is in a nutshell about the point mutation in the ovate gene that we targeted. We developed DNA-based markers. We validated this marker, and this mass we carried out in segregating population to identify better segregants. We have already communicated these results in one of the journals, and we are waiting for the decision from the journal. So if I summarize my entire lecture, then molecular breeding is enhancing the accuracy of selection, which I have discussed throughout in my lecture. It is early identification of desired lines. I don't want to wait for F6 or F7 or F8 to find out the fixed lines. I can genotype them and find out which is the fixed line at a very early generation. There is early generation fixation. Selection of difficult traits. As I have mentioned, we are working on disease resistance of multiple diverse caused by diverse group of pathogens. So it's really very difficult to create epiphytotic condition for a nematode as well as a virus as well as a fungus. So we need to rely on our molecular markers, which are highly robust and diagnostic in their nature. So it allows us to select for difficult traits. Some of the traits like fruit shape, like fruit color, which will be only visible after the fruiting, we can identify them even at seedling stage. So simultaneous selection for multiple number of traits. I want to combine good lycopene content with multiple disease resistance. So this kind of traits which are now it's possible to combine them if you have a very good diagnostic markers in your hand. So early generation fixation of desired alleles, I have already mentioned, and it's a rapid and cost effective manner, because if you take the help of automation, doing genotyping of 300 plants in a single day is highly possible. Even with our facilities, we are doing at least 50, 60, 80 plant DNA isolation in a regular basis, doing their PCR and doing the genotyping. So ultimately, it involves some input costs from chemical and reagents, but it actually reduces the major costs of land, labor, and resources. So these are some of the suggested further readings among these 11. Two is from our group. In one, we have fetched one publication in Plant Cell Reports, where we have identified all the fruit color variant mutant alleles that can be targeted for molecular breeding, to stack them all together to improve the fruit quality in tomato and fruit color. And the second one is regarding identification of this dark green mutant allele, which gives high lycopene content. Both of them have been published in this year 2021 only. So I must acknowledge my team, that is having my colleagues Dr. Shireen Akhtar and Dr. Chandan Roy, and my instrumental students, that is Deepak Maurya doing PhD, Bhagyashree, ex-MSc student who has taken a lead role in initiating the program of molecular breeding, Arnab Mukherjee, my present MSc student. I thank them all because they're instrumental and tireless in their works. I rely on them just like blindly. They guide me what to do, and I'm really thankful for their tireless efforts towards our molecular breeding program. And it will be, uh, if I forget to mention about the contributions, like from Dr. Nagender Rai, who has taken a lead in providing me the tomato genotypes on the basis of which today we are having this molecular breeding program. Dr. Pranab Hajra from BCKV, I thank him for providing very precious materials for fruit quality improvement. And the entire Bihar Agriculture University team, my Honorable Vice Chancellor, Dr. R.K. Shohane, Directorate of Research, Director of Planning, and all other deans and directors, all supporting staffs who has really helped me a lot in uh, giving suggestions and giving all sort of helps and administrative supports. So I thank you all, and I request, our team is now having some of the programs where we are actually inviting application from National Postdoctoral Fellowship. If some PhD students who are going to complete their PhD within 1 or 2 months, I request them to contact me with my email address available in this slide and also in our website, so that he or she can join our lab in this molecular breeding program. And at last but not least, I must not forget to thank our fantastic media center and all the members of the media centers, all the crews of the media centers who work really hard to make this kind of efforts successful. So thank you all. You are all requested to give your suggestions, questions, and queries in the chat boxes so that I can have a chance to reply to you all individually. Thank you. Thank you very much.